Here, another important molecule for Primrose is a vaccine carrier protein called CRM197. And what we wanted to evaluate is whether, incorporating PAZF and doing that conjugation would enable us to develop more precise controlled conjugation for CRM. So we evaluated the CRM197 crystal structure, as well as, again, doing some HDX-MS analysis to see where are there solvent exposed areas where it would be good to insert PAZF for conjugation. CRM197 is used as a carrier protein for like different carbohydrates and things to really boost the immune response to those carbohydrates or polysaccharides. And so we wanted to avoid any published T cell epitopes in CRM. So that was another thing that we were looking at. CRM forms a dimer, also wanted to avoid the dimer interface because we didn't want to disrupt that. So we identified 11 sites for conjugation, did the cloning, inserted amber codons there, and ran this in our PAZF Incorporation expression hosts. And on the left-hand side here, you can see at the 2-liter scale, so this is in high cell density fermentation, we're evaluating what does our titer look like compared to our canonical or wild type CRM strain, which here is shown as control. And you can see that in 10 out of the 11 places where we inserted PAZF, we got really nice expression, at least, or in some cases greater than what we see typically with CRM197 expression in Pseudomonas fluorescens. Of course, we wanted to make sure that the protein looked as we expected. So if we purify these different variants with PAZF incorporated at different sites, we can see that intact mass shows us that we've got a single site of incorporation, and PAZF is inserted where we expect it to be. If we then do the SPAC reaction, we can show that it's functional. We can click different molecules to CRM197. So, of course, our next steps now are to evaluate immunogenicity of the carrier protein with these different insertion sites to see whether these would be good candidates for further development.